90 participants already. So I will start the introduction. It's really a pleasure to have Andre Fenton speak at the JSN today. As I was telling Andre, it is sort of a challenge to introduce him because he just has done so many things, so many different things. And so, yeah, let me tell you about it. Andre grew up in Canada, although he has very strong roots in the Caribbean. He moved to New York in the 90s, and though he sort of made trips here and there, eventually he basically launched his career in New York. He graduated from McGill in Montreal and then did his PhD at SUNY Brooklyn with Robert Muller. Eventually did research in the Czech Republic with Jan Buris, and I guess became faculty or sort of a lab PI at the Institute of Physiology in Prague, and then eventually moved to NYU in 2000, where he was officially an assistant professor, moved quickly through the ranks, and has been a full professor for the last decade at NYU. Of course, you all know him as a neuroscientist for good reason. He's made amazing discoveries, including the co-discovery with Todd Sachter of the role of protein kinase M-zeta, PKM-zeta, in LTP and learning and memory, but has done a lot of other work in spatial navigation of mice, role of place cells in the hippocampus, and also work in autism and in Fragile X syndrome, which is the work that I am most familiar with. But he's just more than a neuroscientist. He's also an inventor. He has 10 patents and counting, including the invention of a rotating arena, which he uses to study place cells in the hippocampus and place avoidance. And I think he'll hopefully show us examples of that. He's also invented a portable EEG device, and even most recently, what I gather is a CPAP type airflow device for patients with COVID that could easily be used in remote locations like Africa and South America. So he's also a phenomenal public speaker. My goodness, he's done more talks than I could ever imagine. But in particular, some of you may have seen him in PBS or in Nat Geo. He's made appearances in This American Life, in Nova Science, and the show Brain Games, which my daughter and I watch on TV sometimes. And even has been a guest in Through the Wormhole, that show that Morgan Freeman hosted. So countless others. And it's not surprising because he's such a charismatic person. You will see once he starts talking, this boundless energy. He's just a captivating individual. And I've only interacted with him on a couple of occasions through the Simons Foundation and most recently through a meeting, virtual meeting. And anyway, it's a delight to have Andre here. He's going to tell us about memory, learning to learn, and the control of cognitive representations. Thank you for coming, Andre.
Well, thank you. Thank you, Carlos, for that very generous and a kind introduction. It's really a joy to be talking with you. So many of you are familiar to me, friends, and the people I've learned from. So this is really very pleasurable for me. Thank you very much for the invitation. Hopefully you can all hear me. And I have sort of out of the corner of my eye, I can see you, but I don't see the chat deliberately because I want you to interrupt me if there are questions. I've prepared my lecture in 3 parts. If I don't get to the last part, that will be fine. So, please interrupt. Let's have as interactive a conversation as we can. I have some disclosures to make. They're not relevant for the talk, but nonetheless, here they are. I'm going to start with a simple movie that I made when I was a graduate student. Hopefully you can hear this. Can you hear the discharge of this place cell?
We can't, no.
Okay.
One way you can do it is if you get out of the sharing, when you try to share again, there's a little button that says at the bottom, audio, sound.
All right, well, it doesn't matter.
Okay.
I'll make the noises for the cell. It's discharging now. Right? Because the rat is in that part of the area of the field where the red dots are shown. And so this is a classic and old-fashioned a demonstration, if you will, that earned John O'Keefe the Nobel Prize in 2014 for his discovery and his insight into seeing those cells as animals explored simple environments and recognizing that they might be signaling places. It was not so obvious. Other people had recorded these cells and seen place cells, in fact, and had not recognized them as such. He shared the Nobel Prize with the Mosers, which, and as you know, they discovered grid cells. And similarly, even the Mosers had discovered grid cells or had recorded them without recognizing that they were grid cells initially. It took them a couple of years to realize what they had in fact discovered. And so what I want to point out is our laboratory and what our aspirations are sort of come from some of the work that's been done in the O'Keefe and Moser lab in the place cell and place cell navigation field. And in particular, what I want to point out is while the award, very well awarded, was for, in some sense, identifying the brain's GPS, as the Nobel Committee put it, what they've really identified is what I like to call the parts list for how you build a navigation system. You can find border cells and directionally tuned cells, distance tuned cells, which are the grid cells, and these place cells. And if you look at a single CA1 neuron, you'll realize that they're receiving multiple kinds of spatial input. And one of the challenges that we have is to try and understand how these cells coordinate that information and actually generate a representation of the world that the animal can actually use. And so our laboratory's overarching ambition, this is not something we've achieved, but it's our ambition to try and understand how you can assemble a coherent, functional, cognitive system from all of these parts, if you will. And so that is the thing that motivates all of the work in our laboratory over the last years. And I'll try and describe for you our approach to that and how things happen. So in the very beginning, when I was a graduate student and I came to Bob Muller's lab, we recorded cells like this, a beautiful place cell. It's discharging in a particular region we call the firing field. And if you were actually at the rig recording these cells, in those days, we had to record them one at a time, and we listened to the action potentials like in the movie that I showed you that you couldn't hear. And we could listen to them, and something remarkable was happening, at least in my opinion. And that is, as I've shown here, if you allow the animal to walk across the firing field of this cell on 2 occasions, in this case, the 2 occasions are separated by 30 or so seconds, and these 2 paths measured at 16 millisecond resolution and 3 centimeter spatial resolution are 97% identical. Okay? And on one occasion, the cell fires exuberantly, a lot of spikes, and on the other occasion, none at all. And that's very unlikely to happen by chance if this tuning curve, in some sense, represents the activity of this cell on any given moment. And that really, annoyed me and bothered me. And Bob Muller kept saying, "Hey, it's biology, Fenton. Biology is messy, it's noisy." But he challenged me to actually go out and prove how unlikely this was, which we happened to do in a few papers, and discovered this phenomena called overdispersion. The fact is that these cells are very unreliable in their discharge in time, although they're very reliable in their spatial tuning. And I've always wondered what that could possibly mean. And you'll see throughout the rest of the talk, I'm always interested in how the dynamics of these cells and trying to understand how it is that they could be useful for understanding where the animal might be or how to recognize a memory, and at the same time be unreliable in this way. I won't dwell on it, but there's an overall idea that drives this research. And we think about, at least the laboratory thinks about these cells as being part of a winner-take-all competitive, if you will, a tracker-type network, where the key thing is that there's mutual inhibition between different modes of firing, between the different functions, if you will, of that the cells can organize themselves into. And there are different ways of conceptualizing this. You can build any toy model, a Hopfield network, and if you provided two inputs, one of them stronger, one will outcompete the other in such a network, driven really by this mutual inhibition. And you'll see this theme come up several times in trying to guide us and understand the biology that I'll describe. So I'm going to start with part one of the talk here. We're going to try and trace really where do we find LTP, where are the long-term persistent synaptic changes that might underlie long-term memory storage in the mouse brain in this case. So the first thing I'll start with is this demonstration that if you induce LTP, long-lasting form of LTP here in the Schaefer collateral, what you can recognize is that LTP induces a number of molecules, but only one molecule seems to be enduring, and it's activated and stays activated a very long time. That molecule is here, PKM zeta. It's a kinase, it's a persistent kinase. The magenta portion here is a peptide inhibitor that we designed. They'll become important in the next slide. What I want to show you here is that if you knock out PKM zeta, our antibodies, our anti-PKM zeta antibodies seem to work well. There's no staining. In a control hippocampus, you can see this kind of expression. But if you were to stimulate here and induce LTP, you see that in the pathways where the fibers, the Schaefer collateral fibers terminate, you induce an increase of PKM zeta expression. This in a hippocampus slice. If you were to take now a fully fledged rat in this case, and we induced LTP under urethane anesthesia here, and we allowed the LTP to last a day. So 22 hours later, we injected ZIP, that inhibitor, which is the PKM zeta inhibitor that's a pseudo substrate. It's actually this little magenta molecule in the model. And ZIP is fairly specific for the catalytic subunit of these atypical PKCs. And what you see is a remarkable thing. After LTP has been established for a day, right, you can reverse it to baseline levels with the inhibitor ZIP. If you were to put the ZIP just on an unpotentiated pathway as shown in the inset here, it doesn't do anything to unpotentiated synapses. It in particular seems to reverse LTP that has been established and is persistent. And for that reason, we think that PKM zeta is crucial for not only the maintenance of LTP, but possibly for memory. And so here's an example of some work some time ago where we used this ZIP to investigate whether it would change place cell firing and change the representations of a familiar environment. And so what I'm going to show you here is the recordings, a summary of recordings from place cells before and after injecting saline, the vehicle, or injecting ZIP into the hippocampus while we recorded the cells. We can record place cells, but more importantly for this slide, we're going to use a Bayesian framework to compute an estimate of where we think the animal is. So we're going to decode the activity that we can record in the hippocampus to identify where we think the animal might be. So we end up with a Bayesian posterior as shown here, and we can estimate how close we are with our estimate from the Bayesian to where the animal actually is. And what's going to be plotted here is the decoding. So if you do this for some ensemble recordings before you inject the ZIP or the saline, you see that we can do fairly well in decoding. Obviously, if you collect more data, you can decode better, at least within limits. So with the second or so of data, we can decode the animal's position far better than chance. What happens after injecting the ZIP? Well, we go to not being able to decode the animal at all. I want to point out the activity, if you look at this paper, is of place cells. They're maintained. If you'd injected the saline, you would do this well. So the ZIP truly seems to erase all memory or all information that had been established before the ZIP was delivered. If you look, the place cells are actually fairly stable during the training session, during the post-ZIP interval, because you can decode using the post-ZIP injection activity, and you can reliably predict where the animal happens to be, at least as well as you can across a couple of hours for saline injected animals. So all of this is just to show that PKM zeta inhibition can lose, if you will, the information that's carried in spike trains. I'm going to come back to that. And maybe this demonstration here in a movie, instead of showing you the data, is really quite striking. We can train the animals in a place avoidance task. Here they are rats. The rats are going to be trained to explore this rotating environment. Right now, it's rotating about 5 times faster than the 1 revolution per minute that is typical. The animal's exploring everywhere. And then what we can do is activate shocks such that when the computer detects the animal in the region that's marked now red, it will electrify the floor, and the animal doesn't like to be in the electrified part of the environment, and it will run away. And you can see after, in this case, 8 training trials, this rat will do a very good job of staying away from the shock zone in this place avoidance task. So now we've established a memory, if you will, and now we can check what happens in this case a day later. I'll show you later, a month later, but a day later we can inject saline into the hippocampus, exactly like we had done to depotentiate the potentiated pathway. And the animal's avoiding very well. I think in this movie, in the next moment, he's going to actually enter the shock zone and run away. He's presumably running away because he's remembered that there's shock there because we're not delivering any shock. If we do the same thing after injecting the ZIP, the PKM zeta inhibitor, you can see that the animal no longer shows any type of avoidance. If you look at this paper, Pashtalkova et al., you'll see that if we were to reactivate the shock zone, the animal would learn to avoid that shock zone fairly normally. It's not, the brain has not been damaged. The animal is perfectly capable of learning. It's just not capable of recollecting that information. And so we've been very interested in understanding how PKM zeta, or how to use PKM zeta to understand where in the brain and how memories might be formed. And remember, PKM zeta is demonstrated to be both necessary and sufficient, actually, for maintaining LTP, at least in the Schaefer collaterals. So we can also ask, where does memory training, here's a version of that task now designed for a mouse, where does this memory training cause persistent LTP? What's shown here in the bottom left is a ARC-driven EYFP signal. So what we've done is, while the animal is expressing the memory, we activate a CRE using the estrogen receptor with tamoxifen, and we activate the expression of EYFP in those neurons that were active during this training. And we can see where the cells are active. You'll notice that the hippocampus is not homogeneously activated in this way. The CA3 region is much less than the CA1 region, the upper blade of the dentate gyrus, much more than the lower blade of the dentate gyrus. And we can do physiology because these different regions receive different inputs, so we can do synaptic domain specific physiology. In this case, I'm showing you what we see when we look at the Schaefer collateral input, that's the CA3 to CA1 input here. If we were to take ex vivo slices after the animals have learned this task, a day later, we can see that that pathway is strengthened in this stimulation intensity versus field EPSP response. And we also attenuate LTP. We can induce LTP, but the level of LTP is actually substantially lower. So this training somehow changes the functional relationships, at least in that part of the circuit. What's remarkable is these changes can last at least a month. And so in this case, it's the same experiment where we train the animals to do this avoidance. We're going to compare the animals in this case to a control group that has never been shocked and is on the arena for the same amount of time. You can see its tracks, it goes everywhere. And after the training, we wait 30 days. The animals are all in their home cage. And then we bring them back, test the retention, again, with no shock. So for about a month, these two groups of animals have had the same life experience. And when we bring them back and test for retention, we can see that the animals that were trained obviously remember. But if you look at the distribution, some of those animals remember very well. the shock zone, and it takes them, 6 or so minutes before they enter the shock zone. Whereas some animals who were trained in red here are no different than the control animals who didn't have to avoid. So some have retained the memory, some have not, and we can divide them into those groups. And now if we look at the animals that had retained the memories and those that didn't, those that did have this potentiated pathway into stratum radiatum. Those that didn't retain the memory are no different from control. Similar with the plasticity adjustment, the animals that remembered have this attenuated potentiation in response to high frequency stimulation, but not the animals that didn't remember. And this is a month later. So we've been very interested in how the brain is changing in this remote, if you will, maintenance of memory. Not surprisingly, we would use PKM zeta to look and see where in the brain is PKM zeta expressed in this elevated way, a day, in this case, 30 days, another month after training. And you can see here, we're going to focus on the hippocampus, but in the overlying cortex, retrosplenial cortex and somatosensory cortex, in the deep layers, PKM zeta is also upregulated in its expression. If you look just in the CA1 region, and you can quantify it here, you can see that PKM zeta is expressed, and this again, 1 month after training, in the pyramidal cell layer, stratum radiatum, also in lacunosum moleculae, at least in these conditions. So Alexandro in the laboratory, he came just for the start of COVID. Congratulations to him. And so he came to do some physiology work, but because of the various pandemic shutdowns, instead decided to do immunohistochemistry and look to see in these brains after this kind of training a month later, where is it that PKM zeta is expressed, but in particular in these green neurons that are expressing EYFP because they were activated when after the animal had learned the task, but when it was challenged to recollect the task on the last day of training. And you can see that PKM zeta is expressed. It's at the spines, in particular in these EYFP expressing cells. But we're going to look, and Alejandro is going to quantify where in these memory associated cells, in particular, the PKM zeta is upregulated and expressed. Again,
let me ask you a quick question, Andre. So
sure,
you mentioned this before, but if you are looking for PKM zeta expression in animals that have just been randomly shocked, but there was no training involved, there's definitely no PKM zeta upregulation, right?
Yes, we don't see that upregulation. We actually do that control, but for simplicity, I'm not showing it.
Yeah, okay.
It's a yoked control. And in what I'm going to show you, I'm just gonna show you the data from untrained animals who run around the same amount. They don't get shocked, and that's what we see. I should also just point out, thank you for thinking about it. Our shock is very mild. In fact, when we measure QORT levels in untrained animals, trained animals, or these yoked trained animals, we can't tell the difference after the first 10 minutes session. The animals do a pretty good job of avoiding in the first place, but even the yoked controls, they don't get shocked very much, and they don't show any kind of freezing or even a stress response, which may help you understand why it looks like baseline. Okay, so what we can see here is that, not unlike the physiology, in the stratum radiatum compartment here in the middle panel, what we see is an upregulation of the EYFP activated cells. That's not surprising. The upregulation of the PKM zeta, that when you merge the two signals, there's a co-expression, and you can see that in particular, it's the EYFP cells that now express the PKM zeta. That is where the increase, the converse, the PKM zeta cells that now express EYFP, that actually doesn't change. So this is exactly what you would predict quantitatively if it was the green cells, the ones that were activated with the memory, that were now expressing the PKM zeta. And that's what we see in stratum radiatum, as well as in the pyramidal cell layer, but we don't see that in lacunosum moleculae after this kind of training a month later.
Andre, David Glansman has a question.
Please, David. Hi.
It works better when you
Hi, Andre. So it looks like the biggest difference is in the cell bodies.
Yep.
So how do you interpret that?
So there are two things. We know that PKM zeta has a role to play, and maybe an epigenetic role to play, in actual transcription regulation. And we know that one of the things that this kind of training does and is necessary is actually in the expression and the manufacture of particular variants of ribosomal RNAs. So there's definitely some nucleus-based regulation that happens. What I'll be able to, I think, what I will argue is, for the purposes of this talk, I'm going to focus on these dendritic compartments because we're going to do physiology that is related to those compartments. There's a whole set of metabolic changes that I think PKM zeta is involved with that I will not address. Why do I think that they're involved with that? Well, because we've done transcriptomics on these kinds of cells in these kinds of experiments, as well as proteomics, and we can see that it's involved in the brain naturally is not making only this change, it seems to make a variety of changes. And that's one of the things I'd like to discuss with you, David. I remember we have to rescue synapses for having a role in memory. They also have a role in managing metabolism.
Okay, just real quick question. Have you published the epigenetic stuff yet?
There is a paper, yes. Todd and another group has published the first indication of that.
Okay,
Thank
you. Thanks a lot, Andrew.
Cool, this is great. Please keep asking questions. So I'm going to summarize this first piece because what I really wanted to be able to show you is that the brain changes in some way physiologically and maybe metabolically, at least in the biochemistry, when memories are stored for a long time, at least after training in this active place avoidance memory. What we can see is that there's a persistent strengthening, if you will, of the CA3 to CA1 synapse, but not in this temporoharmonic synapse to stratum lacunosum moleculare. What we see is this persistently enhanced PKM zeta expression in these corresponding domains. And if you will, these, I would call them traces, if you will, of this cognitive training. They grossly correspond to those of you who are place cell aficionados and so on, to some of our ideas of how the physiology is mediated in the hippocampus, where the CA3 input to CA1 is thought to be more mnemonic or memory bearing, if you will, whereas the temporoharmonic entorhinal cortex of CA1 input seems to be more involved in encoding, if you will, and not in particular for memory. Keep that in mind because I'm going to now focus on some of the physiological changes that underlie what I've outlined here. So let me jump to the second part. I'm going to talk about learning to learn. I'll describe what that actually means in a moment and try and show you that not only were these gross changes observed, but we could find persistently modified, fairly specific excitatory inhibitory subcircuit changes, in particular in the perforant path to the dentate gyrus synapse. And this work was Aing Chung's thesis, joined by Claudia Zhou and Dino Dvorak. Dino's a postdoc in the lab and Claudia a new graduate student. So I'll start with, how did we think about this? So I want to point out that, early in developing this place avoidance task that you see here in the right, that's the rotating arena overhead view, we understood that it was extremely sensitive to hippocampus function. It's the one task that you can make a partial hippocampal lesion and get very clear deficits. And so we understood that it was beyond the demand to simply learn the location of the shock. And with behavioral and various hippocampal inactivation experiments, we were able to show that a key in this task was that the animals also had to learn to actively ignore places on the environment, on the arena. In particular, they had to ignore where they were on the arena surface when they were shot. And so we could see this actually directly in the discharge of hippocampal cells. And what those cells did, and I'm showing you an example here, is they toggled between representing locations on the rotating surface and locations in the room. The same cells would switch as an ensemble, signaling one or the other. In this case, I'm using one of the UCLA mini scopes here to record a group of cells, and we can decode the activity of these cells and represent how much information this ensemble is signaling during a 300 millisecond period about the animal's location in the room or the animal's location on the arena surface. And what you can see here is that the activity, the ensemble activity, toggles between representing room locations and representing arena locations here in blue. And this toggling is not random. In fact, if we look carefully, we can see in these plots down below, which shows the propensity to signal room preferring information. So if you look at the animal's locations in the room during pre-training, there's no particular place that this information is more expressed. But as you train the animal and as you ask it to remember and change the location of the shot zone, you see that the activity preferentially represents where the animal is when it's close to the room defined shot zone, and you don't see that organization on the arena frame, the rotating frame. And in some sense, this dynamic, if you will, functional grouping of the neural activity is, truly direct evidence that there's something like cognitive control of the hippocampal discharge. The animals can know where they are in the room. They can know where they are in the arena. Knowing where they are in the room is important. Knowing where they happen to be on the arena turns out not to be important for avoiding shock. And the hippocampal representation is switching between these two possibilities. If you look a little more closely at that plot, what you can see is that the hippocampal representation prefers to signal locations in the room, in red here, in particular, when the animal is very close to the shock zone, or in the second case marked in gray, as the animal's approaching the shock zone here in the gray, in the red line, and is going to turn and walk away. So the hippocampal activity is toggling between these two, if you will, streams of information about locations in the room that are stationary and rotations that are on the arena and rotating. And in thinking about this, Ayn, when she joined the lab, was able to notice something intriguing about this possibility. So if we were to consider this paper, this is what I pointed out, that was in Nature, where elderly subjects were given a game to play in which they had to use cognitive control. I won't describe why. It seemed to have long-lasting cognitive improvements in this elderly population. And so I asked, why does this work? Why does playing this particular kind of cognitive control video game actually have long-lasting effects, pro-cognitive effects in these aged humans? And I said, well, I don't know, but as a meditator, what I spend a lot of my time doing is trying to my mind, if you will, to ignore or to avoid distraction. And so we wondered whether this had a feature to play here. And so we decided to investigate whether this ability to actually ignore where the animals were on the rotating platform actually was crucial in making some of these long-lasting changes, the kinds of changes that you might find in a cognitive behavioral program like what was used for these aged individuals. So we designed a task using the place avoidance task. So what I'm showing you here in this picture, sort of tongue-in-cheek, there are really three task variants here. We wanted to set up a cognitive task. We had a cognitive control task, but we needed control tasks. And what I want you to appreciate is the control task and the cognitive control task that I'm going to use that I've already showed you are almost identical. They differ in a very, very slight way. So the task I've shown you the video of is cartooned here. I'm just going to change the name to cognitive control training. The animals are on this rotating arena. They have to understand or recognize where they are in the room to avoid shock. And if you will, ignore where they happen to be on the floor actively so as to not try part of the floor where they were shocked. Since the floor is rotating, that is a distracting or a useless thing to do. We used a control task where it looks fairly identical in this picture. What you can't tell is that there's a thin layer of water on the arena surface, which is making those arena cues undetectable. And so the hippocampus here is signaling locations simply in the room, and you can't, the animal can't read location on the local frame of that's rotating. And in this control, the spatial exploration group or untrained group, they're simply on the arena, and it's there's no shock or water. What again is very important to recognize is that in a 600-second, 10-minute session, the rat gets shocked somewhere between 0 and say 6 times, right? Which on the top end, since the shock is only half a second, is about 3 seconds. So of the animal's time in the arena, in these 3 conditions, they are identical 99.5% of the time. The world is physically identical for these animals. The only difference being when there is an active shock, which lasts about half a second, and in aggregate only lasts in total about 3 or so seconds with training. So we're going to train animals in these 3 different ways, and we're going to design an experiment as described here. And so what we are going to do is train the animals so that they learn this task very well. And we're going to wait 1 week and test their retention. Retention is tested simply by letting the animal walk around without shock and seeing how much they avoid. We're going to wait 3 days and train them again in a different room on a different version of the task. And the different version of the task is this cognitive control task I've shown you already. It's in a different room, so none of the cues are relevant. And we're going to look, and what we noticed, and Ein was pointed out to me, is if you compare the animal's initial learning curve shown in blue to its learning curve in the subsequent, in the new environment, the animals learn way, way faster than in the than they had originally. And so they start out not knowing, but their learning is much, much more rapid, and they're capable of avoiding subsequently. If you look at this initial training at the first trial in this new room, you can also see that the animals, depending on their history, learn the task very well. If you look at the number of entrances, it's much fewer for the animals that had the cognitive control training than the animals that had the place learning or no training at all. If you measure the time to first enter, it elevates much higher and faster than it did in the other 2 groups. If you just compare again the time to first enter in the first training session compared to the subsequent training session of these cognitive controlled trained animals, you can see again that they learn much faster in the second case compared to the first. So that may or not be impressive to you, but what if you just took a completely different task in a yet third different room? Here it was a T-maze alternation. We taught the animals to go first to the left and then to the right. If they had this place avoidance training initially, then a week later or 10 or so days later, we tested them in this go left, then go right task. And you can see that initially, first to go left, they're just like the other animals, then go right, they're just like the other animals. But once you actually ask them to reverse the third time and subsequently, they're much quicker at being able to get that where they get shocked for the first time to go to the opposite, if you will, much more than animals that didn't have this training. So we were very interested to understand, what does this kind of training do? How does the hippocampus actually change or the rest of the brain, how does it change to enable these animals to become better at learning arbitrary things, or at least a subset of arbitrary things. And it can't simply be remembering the information or the specifics of the particular environment that they had been trained in. And so we first, wanting to know where to look at, we looked at PKM zeta after this training, and we could see that it was in particular increased in the upper blade, in the suprapyramidal blade of the dentate gyrus, and in particular in these animals that had this training, not like the other animals. And so we're going to focus on the dentate gyrus. This is just quantification of that. And we can also look to see, and this was really informative to us, that if you look at the interneurons, what you see is that in particular, it's the somatostatin expressing interneurons in the hilus that in particular seem to express an upregulation of PKM zeta. So something was going on with these inhibitory neurons, and that can be quantified here. So we decided to study this pathway from the entorhinal cortex, the medial entorhinal cortex, or the medial perforant path, into the dentate gyrus. It's a well-segregated pathway. The middle molecular layer can be well identified electrophysiologically. And we use these silicon probe electrodes so that we could manage to record and measure synaptic strength, if you will, of this pathway as the animal learned and after it had learned for out to 2 months. And so if you do that, what you can do is we designed the experiment in this way so that the animals were running in a wheel, just in a regular exercise wheel before each of the training sessions. They were running in that wheel so that they would be in a physiological steady state, if you will, generating theta for those who care about it, but running more or less constantly. And then we could stimulate the medial perforant path and measure the synaptic response or the field EPSP response across the somatodendritic axis here using these multi-electrode shanks. And we're going to focus here on the dentate field potential response. And we can look at it before training, and I'll just show you here for this talk, before the retention test 7 days later. And if you compare the two, what you can see is that there's a change. The slope of the response is actually lower after the training compared to before. And we're going to talk about this synaptic response. We're going to actually measure the synaptic response in the current source density so that we can localize these field potentials to their actual dendritic compartments. And if we do that, what we observe is that the training actually reduces these synaptic responses. So here, if you compare from before the training till after the training in the animals that had this cognitive control training, you get a suppression or a weakening of that input. This is the quantification. If you look at the animals that simply ran around and had the spatial exploration, no conditioning, no change. If anything, the change is an increase. If you look at the animals that had the place learning, again, no change. This change seems to be specific to the animals that had this particular type of training. So this change, this reduction in the perforant path response seems to last at least 60 days. It's a change that's very persistent. And here's the quantification of that. You can even observe this change now that we know to look for it in the spontaneous, not only the evoked activity, but the spontaneous activity. So the dentate gyrus expresses these things called dentate spikes. It's actually something I'll talk about in the third part of my talk if I get to that. And the dentate spike, if you look at the current source density, you can see that it's very similar in where the sinks are, the excitatory or inward currents are, and the sources, the outward currents, corresponding to the medial perforant path stimulation. And if you look at the dentate spike in the suprablade of the dentate gyrus, you can see that after this training, again, the slope is decreased, just like the evoked responses were. But there are no changes in the infrapyramidal blade of the dentate gyrus. And we see that consistently. Changes and effects in the suprablade, but not in the infrablade. For those of you who are perplexed, I was perplexed too. You know, didn't I already show you that this training causes a potentiation of the Schaefer collaterals? Yes, and it also shows, I can also show you that it produces a potentiation of the medial perforant path to dentate gyrus synapse, as shown here, both after the place learning on the arena with water, as well as after this cognitive control training, compared to the animals that just had the spatial exploration. But this, I want to point out, is measured in hippocampal slices taken ex vivo in a dish. And so there's a lot of, and you'll see why that becomes relevant in a few slides. So how do we think about all of this? This is all, you know, in some sense confusing. So very often in my laboratory, we guess. Sometimes we use evidence, like that there were interneurons that seemed to be changed in the original study I showed you of the Schaefer collateral changes. We knew that they were sensitive to manipulation of inhibitory neurons. Sometimes we use theory and guess. We always guess. And so I come back to this winner-take-all competitive network. The way we were thinking about this is perhaps what we've changed is the inhibition and the ability of the excitatory neurons in this feed-forward perforant path to dentate gyrus system to engage those inhibitory neurons and produce a feed-forward suppression or inhibition of the circuit throughput. So that's what we were imagining, and we're going to do some experiments to test that. So we can take a mouse, a GAD2 Cree mouse that will express, here in this case, channel rhodopsin, importantly, in the neurons expressing GAD2, which are mostly inhibitory neurons. And so what we can do is now, again, reassess that perforant path response. This time, we're going to try to see if the activation, there's no training, but if the activation itself of the GABAergic cells will mimic the response. And so that is the experiment that we're going to do. We're going to do this with the silicon probe, and we're going to do this in the in animals that are under urethane anesthesia. And so what we can see is if you take a home cage control animal and you activate the blue light before you stimulate, you get a reduction very similar to what the training had produced. You see the same thing if you had done the animals who had training, but to no conditioning, the spatial exploration group. Not surprisingly, you see the same thing if the animals had the training in the water and had simply learned a place. But this change is occluded, if you will, or attenuated in the cognitive control trained animals, as one might have predicted if this kind of activation, if you will, increase of potentiation was what had occurred. So we can be a little more specific than that. In this experiment, what we're going to do is pulse the blue light to activate inhibition, very local to where we're going to record and the synaptic response. And so what we can see here is that if you coincide the blue light stimulation with the medial perforant path stimulation, there's no particular change. If, however, you precede if you activate inhibition, now unintuitively, what you get is an enhanced response from the perforant path response, as if this was a disinhibition type of response. And in fact, that is what we find. If you activate in the animals that have the cognitive control training, if you activate inhibition just before the stimulation, you get a potentiation of the response. So as a summary in how this seems to be mediated, what we observed after this training in the awake animal, or at least the intact brain, is an overall decreased response, as if there was increased overall inhibition. And we also believe that what we're observing is this feedback, this potentiation onto feedback interneurons that produce a disinhibition of the medial perforant path input. It's relevant that I pointed out that the somatostatin cells that were increased in their PCAM zeta expression were identified because there's a neuron for the hippocampus aficionados called a hip neuron, a hilar perforant path projecting interneuron that is somatostatin positive and actually plays this role. And we're pursuing whether those are the cells that actually are potentiated and responsible for this. So I'll summarize very quickly this little section. This kind of cognitive control training, we believe, we could demonstrate has caused the animals to be able to learn. And that means their hippocampi circuitry, or at least their brain circuitry, has somehow changed in a way so that they can learn new things or different things better, at least within the limited set that we've checked. How that happens is there's a persistent, lasting at least 60 days decrease in the overall medial perforant path throughput to the dentate gyrus. And the way we think about that in the winner-take-all framework is you have, if you will, noisy inputs into the network. After this kind of change, what you get is a dampening, if you will, or an increased overall inhibition that can remove, if you will, some of the noise, and a boost, if you will, through this disinhibition circuit so that the signal, if you will, can be boosted and the noise decreased. This is actually something that we were able to observe also happening and being mediated by adult neurogenesis in a paper we published with Renee. And so this seems to be a general motif that may be occurring as the animals have a cognitive experience in at least this part of the hippocampal pathway. We're exploring other pathways, but we've only focused on the medial perforant path to dentate in this kind of detail. So I'll just summarize for the time being, right? This kind of
Andre, can I ask you about the learn to learn? Do you think it would extend to other types of, aversive shocks like a light pulse, or would it even, could you do food exploration as the second learning part, and it would extend to that?
Yes. So anecdotally, we haven't finished doing those experiments, but yes, we see it in an object recognition task also. And that's why I've come to the inference, if you will, that there has to be some global ability of the animal. And in particular, where we see these change, where we see the animals having learned to learn, it's in tasks where there's, in some sense, the requirement to distinguish between something that is familiar and something that has changed relative to that familiar condition. So if you put two objects that are similar and the animals learn their location, they learn that just fine. They can, if you switch one of the objects for another one, they learn that just fine. But if you dislocate one of the objects, and they have to now recognize, hey, it's still the two objects, but it's not in the place I had learned it initially, that's when you see the learning to learn manifest.
So what I meant was actually a slight play form of that is, so you do foot shock the first time and you do aversive light exposure the second time, or your object the first time and then the foot shock the second time.
So we have not explored that. There's a, if you will, I keep suggesting to the students who want to explore what kinds of training will do this, that I'm actually very excited that we found one thing that can do this, because I'd like to follow up what kinds of changes happened in the brain under that condition. So we simply haven't done it. But what I can say is this is the kind of change that I would predict the initial training would produce if you saw in the hippocampal activity this fluctuating between multiple potential representations. So we use that, I would use that as the predictor of whether the training would be effective or not.
So Andre, just very quickly, the simplest explanation is that if you engage an animal in any hippocampal task, then any other hippocampal task will be acquired faster.
But that didn't happen when we trained the animals in the hippocampal task to avoid, as shown here, the place learning, to avoid the same shock zone in the same environment with water on the floor.
Got it. Good point. Thank you.
right? That was, you know, the obvious first worry I had, or control. In fact, as you might imagine, as the PI, I didn't believe this to be significant. But when we did the controls like that, and remember, it's the pictures make it look different, but it's very, very similar to the animals. We couldn't see that difference. And the physiology shows that it's different in those two cases. So I have about 5 minutes. If you want, I can tell you another story. I'll tell it very quickly about how these, this, in particular, this circuit and the medial perforant path input to the dentate gyrus is actually controlling how the CA1 field is going to express its place cells, and in signaling locations where the animal currently is versus locations that it remembers through this altered circuitry, if you will. But I'm also happy to have a discussion about what we've already talked about. You guys, you can all decide.
I think that if you can do it in 5 minutes, that would be fine.
Okay, I'll go fast. Dino Dvorak did this work. I'm just going to skip him. Here's a summary of what I'm going to describe. In this place avoidance task, the animals can remember where there's a shock, right, without actually going there. And what we observe is that when the animal remembers that there's a shock, we see that those occasions are marked, if you will, by slow gamma oscillations in the hippocampal field potential in CA1. And as shown here, here are 3 place cells. The place cell that has its field near where the shock zone is becomes activated during this slow gamma oscillation, whereas during the fast gamma oscillations, these other cells are active, and the animal happens to be in these local positions. So I'll just show you that. Here's a single example, a single momentary example of this. If you look and say, where is the animal spending its time? This green trace is showing you that. It's down here where the movie is showing you the animal. But during these slow gamma dominance events, these are the events when the slow gamma activity is much more than the mid-frequency gamma. What we observe, and that's shown here in red, is we observe that the cells that have firing fields where the animal currently is, down here, right, they stop their activity, and the cells that have their fields up near the shock zone, which is not where the animal is, become active for about half a second. And so you can see that shown here. If you allow me to decode an ensemble of these cells, I can show you here what's shown in color is the posterior, the Bayesian posterior, our estimate of where the animal might be. So at time 0, the animal is going to turn and walk away from the shock zone. So 6 seconds before, 4 seconds before, we decode locally. 2 seconds before, when we observe slow gamma dominance, you'll see that in a moment, the activity we observe decodes to places near the shock zone. And as the animal runs away from the shock zone, the decoded activity catches up and becomes local again. Here's another example of that. If the animal fails to make an avoidance and actually enters the shock zone, we don't see this, this kind of activity, this non-local activity. And if you look, during the moments of time, they only last half a second or so of these slow gamma dominance moments, and ask, where does the activity in the hippocampus decode to? You see shown here in the hot colors, it decodes to these areas where the animal actually is not going to go. And so we have decided to study how this slow gamma dominance occurs. And there are 2 basic hypotheses in this paper, and the take home is shown in the title here. The dentate spikes actually control this input from the medial perforant path in particular. And so there are 2 hypotheses. One is that this kind of switching between an encoding where the animal happens to be, or recollecting where it might want to think about or remember that it got shocked, or whatever that might be, could be internally controlled within the hippocampal circuitry, or that there's some external control signal The answer is there's an external control signal. It happens to come from a dentate spike. I won't take you through the circuitry of what the origins of a dentate spike are, but there happen to be two types. One is the medial perforant path, discharging very strongly on the middle molecular layer of the dentate gyrus. The other, that's called the type 1, is the lateral entorhinal cortex, discharging synchronously on the outer molecular layer of the dentate gyrus. We can distinguish these using current source density and independent component analysis. And what we can see is that when these dentate spikes occur, again, they only last, if you will, a few hundred milliseconds. We see a very strong increase in the firing rate of neurons that we record in the dentate gyrus, neurons we record in CA3, and a little bit of neurons that we record in the CA1 region. And this doesn't happen with the second type of dentate spike. And I'll show you in particular, and sort of wrap it up here, that when these dentate spikes occur, what we see is a very strong co-firing of CA3 with CA1 neurons. And we've done this with what we believe are monosynaptic identified connections between those cells. This plot shows you on the x-axis the time relative to one of these type 2 dentate spikes, where the medial perforant path is the originator. On the y-axis here is the time from the onset of a CA3 cell discharging. And what you see is in color is the probability of a co-occurrence or simultaneous activation, of a CA1 neuron with that CA3 cell. And you can see that they tend to co-fire within say 10 or so milliseconds, which corresponds to, interestingly, there's a synaptic delay that we also observe from dentate to CA3 to CA1. The details of this we've worked out. What I wanted to simply point out here is that this same pathway that seems to change under the learning to learn conditions and seems to change and persistently signal alterations in how it functions, this also seems to be a pathway that can switch the hippocampus from an encoding mode, if you will, into a recollection or memory mode. And that in particular seems to be the pathway that is being adjusted in these trainings. There's an inhibitory interneuron story that I can tell you about, but I'll just get to the conclusions. Okay, so hopefully not everybody is interested in the hippocampus, but hopefully what I've been able to convince you of is that the hippocampus, the neurons in the hippocampus as a network, it can switch between encoding and recollection, different information processing modes. And it changes the registration of the place cell map, if you will, from a local, from where the animal currently is, to these distal cues there, if you will, representations. And all of this is under the control of this synchronous excitation inhibition discharge events, in particular mediated by the entorhinal cortex or medial entorhinal cortex that I've described. These dentate spikes dramatically, they really very strongly change activity throughout the hippocampus, and in particular the overall hippocampal circuit function. So this becomes something that you would like to actually be able to control if you'd like to change how the animal or an individual is capable of understanding and collecting new information. And the exciting part of this work to me, if I step back and think sort of broadly about it, it seems to suggest that behavior, appropriately curated behavior and cognitive training, it really may have the potential to make these long-lasting, persistent changes in the ability or the functions of the neural circuits. And we have potential access to being able to organize that kind of training to actually change, if you will, sculpt how these circuits might actually function. And maybe that is a way that we should think about trying to manage mental dysfunction and other challenges that people have when these circuits seem to fail them. And all of these changes that I've described, again, they're persistent, they're long lasting, they seem enduring, arise from a few hours of behavior. So with that, I thank you for your attention, and especially the funding agencies here in red who have supported the work I presented, and Dino Dvorak, Eunhye Park, Ein Chung, Basma Radwan, Fraser Sparks, Zoe Talbot, and Milena Van Dyke. These are all the people who contributed to the work that I presented. Thanks for your attention. So weird talking.